good morning. So in today's video, we are going to explain what is the DNA replication. So keep on watching. DNA replication. So what is a DNA replication? So the DNA replication is a process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. While the replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic DNA as the parent cell. So DNA copying, each cell division cell must copy its entire DNA. So each daughter cells gets a complete copy. So the rate of the synthesis, the bacteria equals 1000 bases per second while the mammals is 100 bases per second. So the problem is with a single replication origin in DNA, the bacteria genome 4 times 10^6. So it takes 20 minutes to copy. While the human is 3.2 times 10^9, it would take 10,000 times longer. So the DNA copying solution. So the double helix has to be copied. So semi-conversation solution exists and each daughter cells gets one of the original copies. So in this picture, you could see how the replication works. So the DNA replication is copied by a replication machine that travels along each replication fork. So one of the most important members of this complex is DNA polymerase. So it is able to add DNA subunits making into a new DNA. The DNA polymerase is first discovered in 1956 by Kornberg. Its bacteria is E. coli. So the bacteria have three types, the DNA polymerase 1, 2, and 3. DNA polymerase 3 involved in replication of DNA, and the DNA polymerase 1 involved in repair. So the humans have four types, which is the DNA polymerase alpha, beta, delta, the nuclear DNA. So the DNA polymerase gamma is responsible for mitochondrial DNA. Before we proceed, let us know first what is a DNA polymerase. So a DNA polymerase is a member of a family of enzymes that catalyze the synthesis of DNA molecules from nucleoside triphosphate, the molecular precursors of DNA. So, these enzymes are essential for DNA replication and usually works in groups to create two identical DNA duplexes from a single original DNA duplex. DNA polymerase have two properties: only synthesize DNA in one direction, 5-strand to 3-strand, only add to the end of existing double-stranded DNA. Therefore, they cannot start synthesis of DNA from scratch. RNA polymerases can, but not DNA polymerases. So you can see the direction of the fork movement, the leading strand template of left-hand fork, and the lagging strand template of right-hand fork. So, it is most recently synthesized DNA. So, in the lagging strand template left-hand fork, the leading strand template of right-hand fork. So, what happens at the other strand? So, the 3 to 5 strand replication is solved by Reiji Ogazaki, who invented. He first saw that one strand made in a continuous manner, which is the leading strand, and the other from short discontinuous pieces, which is the lagging strand. So, the discontinuous pieces are known as Okazaki fragments. So, the Okazaki growth. So, the RNA is added by primase 3 to 10 bases. So, the DNA is added by DNA polymerase alpha. So, primase can add RNA synthesis de novo. We have a RNA-DNA joint. So, RNA is involved in eukaryote DNA replication. Now is the closing the gap between Okazaki fragments, which is the RNA primer removed by RNase H. Then the DNA polymerase delta fills the gap. Then the DNA ligase fills the gap closer. Stabilization of the replication machine. So the DNA polymerase delta and epsilon are attached to the DNA and held in place by other proteins. Sliding clamp proteins allow the stable binding of DNA polymerase and strand synthesis. Clamp loading proteins aid in attaching the sliding clamp proteins. So DNA polymerase has a proofreading mechanism built in. It checks for base matching, removes mismatched bases by going backwards. It reasons why it's not able to build DNA in the 3 to 5 direction because it has no energy from ATP hydrolysis. Then it makes just 1 in 10EA or 10E9 bases added. Unwinding of DNA strands by helicases, it requires ATP. So the single-stranded DNA binding proteins bind to single-stranded DNA to stabilize the structure. So the RPA or replication protein A in eukaryotes. So topoisomerase helps with prevention of DNA strand twisting swivels. So the two types is type 1, break one strand only and then rejoin. Then the type 2, break both strand and then rejoin. So as you can see in the picture is the protein aids, the replication fork movement, the helicase, the leading strand, the lagging strand, and the single-stranded DNA binding proteins. So what happens to the histone? So nucleosomes are disruptive during replication. Each DS strand gets halved. New histones added by chromatin assemble factors. Telomerase has an enzyme, reverse transcriptase, acts without DNA template as it has its own RNA template stored inside. So, the DNA is extended each time it is copied. However, other mechanisms restore length in germ cells. Somatic cells do not have enough telomerase to keep this process going, so they lose telomere length, and this is thought to result in cell death.
Caught in the crossfire.